So we're talking about cellular respiration today. And I have a new kind of a question. I have a text question I'm asking you right now. So we'll see how this works. I'm asking you a question. Why do we breathe? On your device, theoretically, you should be able to give me an answer, and your answer should anonymously appear on the screen. So let's see if we can get some student responses. We need to breathe to live. Yes! Absolutely. Gas exchange, to stay alive, to get oxygen, to circulate blood. Wow, they're coming in fast. All right. So I'm learning something right now. Getting text responses with a class this size is a little overwhelming. So we have bring oxygen to stay alive so we can live, so we can live. Excellent. Gas exchange so we can live. Excellent. And this is not a participation-based question. This was just an experiment. I wanted to see how these questions work. So if you don't submit an answer, don't sweat it on this question. So I'm seeing a common theme here. We need to breathe so we can live. If you have a chance, here's a link to a video you should watch outside of class. This video is going to be like the one I linked for on protein synthesis. It's going to take everything we're talking about today and summarizes it in a short, sweet, amazing 4-minute video. So let's talk cellular respiration. Here's the big picture. We take glucose, C6H12O6. That glucose, or blood sugar, which is a monosaccharide. It's a 1 subunit sugar. This glucose is combined with oxygen gas. And then we turn that glucose and oxygen into carbon dioxide, water, and ATP, or cellular energy. Now I want you to note there's an important theme here. And I'm trained as a biochemist. And when biochemists learn about cellular respiration, we get really excited about counting carbon atoms. So that's the approach we're going to take today with cellular respiration. We're going to focus on the carbon atoms. Glucose has 6 carbon atoms. So we have 6 carbons going into the chemical reaction of cellular respiration. And then we have 6 carbon dioxides coming out of cellular respiration. And when we take this glucose, we are quite literally burning it in our bodies. Think of burning wood in a campfire. It's the exact same chemical reaction. But you probably noticed that, like, you don't have bonfires in your bellies. I mean, there's a slight difference. There's some nuances. When we have the bonfire, that is an out-of-control chemical reaction. It's just happening all at once, and it's not in a controlled environment, and we're not harvesting energy from the burning or the oxidation process. In our bodies, when we burn food, when we literally burn our calories, we do it in a more efficient method, or a more efficient process. We use enzymes to control which chemical bonds are oxidized, and as we oxidize individual chemical bonds, we can harvest the energy from it. So when we burn our food, we burn it slowly so that we can harvest the energy to make ATP. And that's a primary or key difference between the bonfires that you enjoy over the summer months and burning food to make energy. Now, as we're going through this process, we need some helpers. The enzymes all by themselves aren't adequate. So we're going to have some transporters to help the enzymes, or some coenzymes. These electron carriers, or coenzymes, function to help us harvest the energy as we burn our sugar, as we burn our food. We call these helpers coenzymes, and they function to carry high energy electrons. As we're slowly burning our food, one covalent bond at a time, we'll harvest highly energized electrons. And we use those highly energized electrons to make the ATP directly. The 2 coenzymes that are the most important for cellular respiration are NAD and FADH. Here's an excellent summary slide for you. Cellular respiration, or making ATP out of glucose, takes place in cells. So we have a big rectangular cell up on the screen right now. The cytosol of the cell is represented in blue. And then we have a mitochondria in gray. And as we look at this slide, you can note that there's 3 key steps that are pumping out ATP. In green, we have glycolysis. In orange, we have the citric acid cycle, which is also referred to as the Kreb cycle, and is also referred to as the tricarboxylic acid cycle. Kreb cycle is the old school name. Citric acid cycle is the name that's most commonly used now. And then in purple, we have the electron transport chain. We take all of these 3 metabolic processes, glycolysis, the citric acid cycle, and the electron transport chain, and we use them together to make ATP. These are the 3 main parts of cellular respiration. So as we look at these 3 main parts, glycolysis takes place in the cytosol, and then the citric acid cycle and the electron transport chain take place in the mitochondria. If you think back to Monday of this week when we talked about organelles of the cell, we mentioned how the mitochondria is the powerhouse of the cell. Its primary function is to make ATP. And when we look at the mitochondria in our diagram, both the Kreb cycle, both the citric acid cycle and the electron transport chain in the mitochondria are pumping out lots of ATP. So let's talk big picture. We're more than just a cytoplasm and a mitochondria. We have multiple organ systems in our bodies that work together. The glucose, the sugar that gets in your blood, doesn't magically appear there. It comes from your digestive system. You eat your sugars to get your food. And we are genetically pre-programmed to crave sugars, to prefer to use sugar as a source of ATP. So we can get blood sugar from our digestive tract. Glucose can go from the digestive tract and be dumped into the bloodstream. From the bloodstream, that glucose can go to the cytoplasm, and then from the cytoplasm, the glucose can be chopped up, sent to the citric acid cycle, and from there, the electrons can be sent to the electron transport chain. And if we think of some tissues in our bodies, particularly our nervous tissue in our brains, our brain needs a constant supply of glucose. But we aren't constantly eating sugar. So to help maintain homeostasis for our blood sugar, we have our liver that helps us out here. You probably ate breakfast this morning, and you're probably not hungry right now. After eating breakfast, assuming you ate breakfast, your blood sugar spikes, and some of that extra blood sugar you store to use later. You take that extra blood sugar, and you can put it in your liver and store it as a polysaccharide known as glycogen. And then usually like 11:30 a.m., 12 a.m., or 12 noon, right before you eat lunch, when it's been a while since you've eaten a meal, your liver is now taking that glycogen, chopping it up, turning it back into glucose, and putting the glucose back into your bloodstream to maintain homeostasis. And then we can take that glucose, put it into the cells, and have ATP generated. So let's talk about step 1, glycolysis. If we had to summarize the purpose of glycolysis in a sentence, the purpose of glycolysis is to make a glucose smaller. We take a glucose molecule and chop it in half. And then we take that smaller piece and we send it into the mitochondria to be processed even more. Now, there's more to glycolysis than just chopping up a glucose molecule. There's a lot more to glycolysis. But the primary function we care the most about in terms of making ATP is that we take a glucose molecule in the cytoplasm, chop it in half, and then send those pieces into the mitochondria. Let's talk about some of the other details. When we look at this glucose molecule, we need to start burning it. This is the sugar that we're literally burning in our cells, within the cytoplasm of our cells. And to start this process of burning it, we need to kickstart the chemical reaction. This is kind of like when you have to start the bonfire in your backyard over those summer months. If you pile the wood in the fire ring, it doesn't just spontaneously catch on fire all by itself. You need to supply some activation energy. Usually it's in the form of some friction from a match or a little bit of a spark. you have to supply a little bit of energy to start the chemical reaction, to start the fire. And our body is no different. We have to supply a little bit of energy to start this process. So in this diagram, energy is increasing as we go up. And we start out with the glucose, and we increase the amount of energy by adding ATP to the glucose. And this process of adding the ATP to the glucose molecule chemically is going to change it. I am not asking you to memorize what all of the in-between molecules are called. And I'm not asking you to memorize what all the in-between enzymes are called or the activation energy requirements of those enzymes. I just want you to know that we start out with glucose. We add energy in the form of ATP to kick-start the reaction. And then after we kick-start that chemical reaction, we will take NAD, one of the electron carriers, and it picks up high-energy electrons. And then we start to make ATP. So we used up 2 ATP in the beginning. We energize the molecule. And then we take away high-energy electrons. And then we take away 4 ATP. And at the end, we have 2 pyruvates, or 2 pyruvic acid molecules. So when we look at glycolysis, we need to emphasize a couple things. It happens in the cytoplasm of the cell. Another key thing we need to emphasize with glycolysis is that it does not require oxygen. We breathe so that we can live. And we do glycolysis. We go through glycolysis so we can live. But this particular part of cellular respiration is anaerobic. And when we say anaerobic, it does not require oxygen. When we look at glycolysis, we start off with a glucose molecule. We end with 2 pyruvic acids. And if we look at the overall net equation, the net yield of ATP is 2. So for those of you who are into accounting or maybe like looking at your paychecks, there's gross and net. The gross yield of ATP was 4. Ultimately, 4 ATP is produced, but since we used 2 ATP to kickstart the chemical reaction, 4 minus 2 gives us a net yield of 2 ATP. And mathematically, if we want to keep track of our numbers here, we have 2 ATP generated per glucose molecule. We need to keep our frame of reference. We're going to focus on one individual glucose molecule with its 6 carbon atoms. So when we do bookkeeping and we talk about numbers of ATP and numbers of carbon dioxide, this is all relative to a starting point of one glucose molecule. Here's the chemical pathways involved. We start off with glucose, go to glucose 6-phosphate, fructose 6-phosphate, and a whole bunch of other chemical intermediates. Again, don't memorize them. This is just to show you or to emphasize to you the one glucose is chopped in half and turned into 2 identical pyruvic acid molecules at the end. Now, if we take a 6-carbon glucose molecule and we chop it in half, those pyruvic acid molecules that are left over at the end should have 3 carbons each. 3 plus 3 giving us 6 carbons. So we're going to keep track of the carbons. We're going to do our bookkeeping in terms of the carbons involved. So glycolysis is an anaerobic process. It does not require any oxygen. And when we look at glycolysis, it really doesn't make that much ATP per glucose molecule. 2 ATP per glucose is the net yield of glycolysis. And if we had to rely on just glycolysis to stay alive, well, we wouldn't stay alive very long. Our energy needs are much higher than 2 ATP per glucose. Medically speaking, when you're looking at pathogens that are able to use oxygen can make more ATP. They can go through the aerobic respiration pathways, and because they make more ATP, they can reproduce and multiply and spread throughout the human host quicker. Pathogens that cannot use oxygen, that rely on only anaerobic respiration, have a slower life cycle because they generate less ATP with that less efficient process. So aerobic uses air or oxygen and makes more ATP. Anaerobic does not use air or oxygen and makes less ATP. So let's focus on anaerobic respiration, also known as anaerobic fermentation. In human beings, if we are in a low oxygen environment, so maybe you're walking up the steps to go from lower campus to upper campus to get back to the dormitories. And by the time you get to the top of those stairs, you're just breathing really hard because you just walked up, I don't know, 20 flights of stairs. You're in a low oxygen environment. And it's not that there's less oxygen in the atmosphere around you. It's that the skeletal muscle cells of your body are suffering from a less than ideal concentration of oxygen. They don't have enough oxygen to go through exclusively aerobic respiration, and they shift metabolic pathways to anaerobic respiration. Now, in human beings, if we don't have that oxygen, we can't go from glycolysis to the citric acid cycle. And instead, we change directions, we shift gears, and focus on fermentation. And this fermentation process in human beings is going to focus on removing electrons from the NADH. That's the primary purpose of this fermentation process. If we can offload the electrons from the NADH, we can regenerate the NAD, and they continue going through glycolysis. We can continue limping along, producing low concentrations of ATP with this inefficient process. In human beings, to get rid of the electrons from the NADH, in fermentation or anaerobic respiration, we take pyruvic acid and turn it into lactic acid. So you should circle this lactate and put an arrow to it. Lactate is also known as lactic acid. When we don't have enough oxygen in our cells, the pyruvic acid from glycolysis is turned into lactic acid. And then you have the lactic acid buildup in your cells. Now, lactic acid, well, it's an acid. It screws with the pH of your cells. And we need to get rid of the lactic acid from our cells to maintain homeostasis relative to our pH in the cells. It wasn't me that time, right?
No, I didn't realize it was there.
Oh, okay. So we take that lactic acid that's in our cells. So let's think back to going from lower campus to upper campus. You're walking up 20 flights of stairs. Your muscles are starting to burn. You're feeling the burn. And there's too much lactic acid in your muscle cells. Your muscle cells will take that lactic acid and get rid of it via diffusion. Lactic acid diffuses out of the cell with its concentration gradient, goes into the bloodstream, and heads to the liver. And then your liver will take the lactic acid and turn it back into pyruvic acid by oxidizing it. This is one of the primary reasons why when you're done exercising, you're still huffing and puffing. You've built up a lot of lactic acid in your body. That lactic acid is in your liver, and your liver is trying to process it to get rid of the extra lactic acid you built up. And to get rid of that extra lactic acid, your liver is attaching oxygens to it. It oxidizes it. And to get the oxygen, you have to breathe really hard. So even though you may be done running the 100-meter dash, you're going to huff and puff for a couple minutes afterwards to get enough oxygen into your body so your liver can take the lactic acid and turn it back to pyruvic acid, which is eventually sent back to your cells and then funneled back into the citric acid cycle. So our liver takes lactic acid and turns it back into glucose, more specifically glucose 6-phosphate. And then we take that glucose intermediate and we turn it into glycogen. So once the lactic acid gets to the liver processes it, turns it into a kind of glucose, and then takes that glucose and makes a polysaccharide of it, glycogen, and we can actually store the sugar in our livers. This is one of the reasons why the liver is an organ that you can still buy at the grocery store. It has a lot of carbohydrates in it, and out of all the meaty parts of the body, the liver is one of the ones that we as humans still prefer to eat. Skeletal muscle cells also have a lot of carbohydrates stored in them, which is one of the reasons why we also eat skeletal muscle tissue. Think of a hamburger or a steak. Now, when we look at anaerobic fermentation, it's not very efficient. We don't make a lot of ATP through this process. It's pretty wasteful. The lactate that's produced is toxic, and we need to process it quickly. If we don't process that lactate in a timely fashion, we lose homeostasis based on the pH, and then we lose the ability for our cells to function properly. So let's think of skeletal muscle cells. If we have too much acid buildup in the skeletal muscle cells, the ability of that cell to contract is reduced, and it becomes harder to do physical work because of that loss of homeostasis. I need to emphasize to you, our brains have no anaerobic fermentation. Our brain is kind of the diva of the body. It only finds aerobic respiration acceptable to make its ATP. And it will use sugar only, please. We don't use the other macromolecules to make ATP with our brains. And this is one of the reasons why, if you're hungry, one of the first parts of your body that starts to malfunction is your brain. You probably noticed if you're really hungry, it's really hard to learn. This is one of the reasons why the Department of Education gives breakfast in public schools, because there's lots of research that shows that if you eat a meal, your blood sugar goes up and your brain functions properly. So let's hack your bodies. Make sure you eat breakfast. Don't skip your meals. If you're hungry and you're trying to study, eat a snack and then keep studying. Let's go back to aerobic respiration. During aerobic respiration, we have oxygen. So instead of taking that lactic acid or the pyruvic acid from glycolysis and turning it into lactic acid, we'll take that pyruvic acid and send it to the mitochondria. So in glycolysis, we take a glucose, chop it in half, and turn it into 2 pyruvic acid molecules. If we have oxygen present, those pyruvic acid molecules are going to be sent to the mitochondria. Pyruvic acid, or pyruvate, has 1, 2, 3 carbons in it. And the process of taking this 3-carbon acid and sending it into the mitochondria causes us to lose our first carbon dioxide. So we've gone from 3 carbons to 2 carbons. We have 2 red carbons at the beginning, and we'll have 2 red carbons at the end. Pyruvic acid, in and of itself, chemically can't be sent to the citric acid cycle. So instead, we have to convert it to a different molecule called acetyl-CoA. And that acetyl-CoA is what we dump into the citric acid cycle inside of the mitochondria. So let's review carbons again. We went from 6 carbons to 3 carbons, down to 2 carbons. And now we're entering the citric acid cycle. If I had to summarize the citric acid cycle for you in a sentence, the primary function of the citric acid cycle is to take high energy electrons and send them to the electron transport chain. Because really, I'm a very ATP-centered kind of a guy. I really care about making ATP. And if we only are focusing on making ATP, the primary function of our citric acid cycle is to harvest those high energy electrons and dump them into the electron transport chain. Now, as a caveat, our citric acid cycle is the roundabout of metabolic pathways. Quite literally, it's a circle, and we have hundreds of metabolic pathways that enter into the citric acid cycle. This is how we can take a protein molecule, break it apart, and turn it into a fat molecule. We can take fat and turn it into sugar. We can take sugar and turn it into fat. We interconvert between the macromolecules using the citric acid cycle as the linchpin, the central part of these metabolic pathways. And we are not covering them in detail, and I'm not asking you to memorize them. So what I want you to know is that this citric acid cycle has a lot of exciting things happening in it in terms of fat metabolism, steroid metabolism. It's the central hub in the human cell for metabolic pathways. We're focusing on ATP generation, though. So, from an ATP point of view, our citric acid cycle is going to happen inside of the liquid of the mitochondria. That liquid on the inside of the mitochondria is known as the mitochondrial matrix. And as we look at this process, I'm skipping ahead here, we take the acetyl-CoA, and let's see if I can scribble on the screen here. It worked last class period. Let's go with red this time. The acetyl-CoA is dumped into the citric acid cycle and turned into the first molecule of the citric acid cycle called, appropriately, citric acid. This is why the name citric acid cycle is becoming a more popular name than the Kreb cycle. Kreb is based off the last name of the gentleman who discovered most of these metabolic pathways. And, don't get me wrong, he was a very intelligent man for doing that. But in terms of making it easier to memorize the pathway, calling it a citric acid cycle is more intuitive. So we take the acetyl-CoA and the 3 carbons here from the glucose and dump them into the citric acid cycle. And as we're going through the cyclic process, we take an electron and put it on an NADH. We make a carbon dioxide. So we just lose a carbon right there as a carbon dioxide. We generate another electron carrier with NADH. We generate an ATP molecule. We generate an FADH2, so we put another high-energy electron into an electron carrier and harvest it. And then we harvest another electron and generate another NADH. And I'm missing it. We have one. There's a carbon dioxide there. There's our second carbon dioxide. We dump 2 carbons in, so 2 carbons have to come out as 2 carbon dioxide molecules. So those are the highlights of the citric acid cycle. We take that acetyl-CoA and we chop it up, remove high-energy electrons, and make carbon dioxide. So, pull out your device. I have a question for your learning pleasure. Let's say you're hitting the gym and you're burning up some body fat. How do the atoms in the body fat leave your body? When you lose weight and you go from weighing 150 pounds to 140 pounds, you had to lose something physically from your body. It's not like you chop off a hand. Where does the fat go when you burn body fat and lose body fat? How does it leave your body? Do you sweat it out, urinate it out, defecate it out? Just have it converted to heat, take energy, matter and convert it to energy, or do you breathe out the atoms? Think of the law of conservation of mass. Matter is not created or destroyed. How do our love handles leave our bodies? They don't just walk away. The correct answer is that we breathe our love handles off. Yeah, let's talk about this one some more. For the record, most students get that question wrong. So let's go back to the citric acid cycle. Our citric acid cycle is the metabolic hub in our bodies. We use the citric acid cycle to take fat and convert it to sugar, sugar convert it to fat. It's where we interconvert things. And when we process body fat and dump it into the citric acid cycle, we generate those carbon dioxide molecules. And those carbon dioxide molecules are physically how we remove the carbons from the triglycerides in our bodies. Those carbon dioxides diffuse into the cytoplasm, then diffuse into the extracellular fluid. They go to the blood. From the blood, they go to the lungs, and then you exhale them out. This is why when you're burning lots of calories, you breathe harder because you have extra carbon dioxide you need to get rid of. And for the record, sitting on the couch watching Netflix and just breathing in and out quickly doesn't make you burn fat quicker. You have to physically rip it apart and turn it into ATP and carbon dioxide. Hyperventilating will just make you pass out. So let's talk about the electron transport chain and oxidative phosphorylation. Our electron transport chain has one primary job: make the ATP. It does a lot of other things, but more than anything else, it makes lots of ATP in our bodies. It also happens in the mitochondria. And instead of taking place in the mitochondrial matrix, the electron transport chain is going to take place on the inner membrane, on the inside of the mitochondria, the cristae of the mitochondria. And if you look at an electron micrograph of the mitochondria, that inner membrane folds back and forth to increase the surface area to maximize the amount of ATP we can make with the electron transport chain. The NADs and the FADs from glycolysis and the citric acid cycle that have been harvesting their high energy electrons are going to dump them off at the electron transport chain. As the electrons are dumped off at the electron transport chain, they bounce from protein to protein to protein in the chain of proteins, hence the name electron transport chain. It's a series of proteins that transports electrons between them. As these electrons are transported or shuttled in between proteins, they lose energy. And as they lose energy, they go from being a high energy electron to a very low energy electron. And as they lose energy, the energy that they lose is used to make ATP, adenosine triphosphate, out of adenosine diphosphate. It's used to add a phosphate to an ADP. So, I do not want you to memorize the proteins. I don't want you to memorize the individual cofactors, which is why I really like this diagram, because it's pretty generic. We have a NAD right here and a FAD dropping off an electron. The electrons bounce between the three proteins. As they bounce between the three proteins, they lose energy. The energy that they use will take protons or hydrogen ions and move them against their concentration gradient and concentrate the hydrogen ions in the inner membrane space, in between the outer and inside membranes of the mitochondria. And they build up a concentration gradient of hydrogen ions. That concentration gradient of hydrogen ions will then flow downhill with the concentration gradient through a special enzyme called ATP synthase. As the name implies, ATP synthase synthesizes ATP. Now, I asked you at the beginning of the class, why do we breathe? I need to emphasize this molecule right here. There is our oxygen. We breathe so we can have oxygen to receive the electrons. Think back to your chemistry classes. Oxygen is highly electronegative. It has a tendency to receive electrons, which makes it the terminal electron acceptor in the electron transport chain. So as these electrons are bouncing from protein to protein to protein, eventually they attach to an oxygen molecule or an oxygen atom, and then that oxygen is transformed into water. If you want to know more of the nitty-gritty details, here's a slide showing you the intermediates, including the protein carriers. You do not need to have this memorized. This is mostly an FYI. I forgot to emphasize the skull and crossbones right here. When somebody is poisoned with cyanide, cyanide causes a traffic jam in the electron transport chain and makes it so that you no longer can move electrons through the electron transport chain. And if you shut down the electron transport chain with a cyanide ion, then you stop generating the hydrogen ion concentration gradient, which makes ATP synthase stop working, which means you stop generating lots of ATP via aerobic respiration in the presence of cyanide. I've been told, and don't quote me on this, but I believe for about 1/3 of the U.S. population, apparently cyanide gas smells like almonds, I think. It's been a while since I've read up on cyanide poisoning. So if you walk into a chemistry lab and you smell almond, you should probably just walk out. Unless it's organic chemistry and you're synthesizing that particular ester in your organic chemistry lab. So when we think of cyanide, it clogs up the electron transport chain, and by causing the electrons to stop moving, cyanide makes it so we stop making the ATP that we need, and that's how cyanide kills us. The electron transport chain, to emphasize this, is going to pump lots of protons or hydrogens into the intermembrane space. As those hydrogens are going to the intermembrane space, they flow with their concentration gradient into ATP synthase, causing it to physically spin like the turbine on a dam. And as ATP synthase is physically spinning, it will take ADP, adenosine diphosphate, and smash it into a phosphate molecule and turn it into ATP. So when we look at the big picture, we make ATP out of sugar 2 ways in our body. We can take glucose and turn it into ATP 2 ways. We have our direct substrate level method. This is what we do in the glycolysis, where we're breaking apart that molecule, that glucose, and turning it into pyruvic acid. And during this process, we generate ATP directly. We also make ATP directly in the citric acid cycle as well. Another way that we make ATP is with this oxidative phosphorylation. In other words, we use the proton gradient in the electron transport chain to cause ATP synthase to spin. Using the electron transport chain to power ATP synthase, or to have oxidative phosphorylation, makes most of our ATP. So if we think of it in terms of a per glucose molecule, we make 32 to 34 net ATP per glucose molecule with aerobic respiration. So, guys, gals, we have a review question. Please match the metabolic pathway with the correct response. Does glycolysis best match with A, B, C, or D? So on and so forth. This is kind of a beefy question. I'll give you some more time. So we have about a minute left. Does glycolysis pair with turns oxygen into water? Think about the big picture slides I gave you. What's the main purpose of glycolysis? Main purpose of the citric acid cycle? Main purpose of the electron transport chain? So we're down to about 15 seconds. Five seconds. Please make sure to answer before I give you the answers. So when we look at glycolysis, glycolysis breaks apart glucose. It does not need oxygen. When we look at the citric acid cycle, it does need oxygen, and it's going to send high energy electrons to the electron transport chain. When we look at the electron transport chain, it is going to take oxygen, turn it into water, and it makes a ton of ATP. And when we look at lactic acid fermentation, it does not need oxygen. and we use it to get rid of pyruvic acid. Yeah, I believe 2 more slides, maybe 3. I'll go quick. When we look at glycogen metabolism, we can take glycogen and make it in our liver. There's a special name for this metabolic pathway, taking glucose and turning it into glycogen in our liver. It's called glycogenesis. And when you see this term, I want you to look at the root words, and the root words overlap with each other. First, we have glycogen. This metabolic pathway affects glycogen, and what does it do? It makes or creates glycogen. So the second root word is genesis, and the suffix. And when we think of genesis, and we look at the Latin roots for genesis, in Latin, genesis means to create. So glycogenesis is the process of creating glycogen. We also have glycogenolysis. Here's the root word for glycogen. And we lyse it via hydrolysis. We take a glycogen molecule and chop it up and turn it back into glucose. And then the last metabolic pathway I want to emphasize to you relative to carbohydrate metabolism is gluconeogenesis. What happens during gluconeogenesis? We make new glucose. Neo is a root word that means new. So during gluconeogenesis, we can take a protein and turn it into sugar, or we take a fat and turn it into sugar. This is making sugar out of something that was not a carbohydrate. Here's a summary slide that's the same content that we had before. We take blood sugar, we chop it up into pyruvic acid, or we can turn it into glycogen in the liver, and we're done with our content. Make sure to read the announcement about the exam this weekend on Canvas. Email me any questions you have over the weekend.